Okay, thanks for coming back, Teams friends. That was good timing. I am recording this, and if at any time you can't see what I'm doing, just tell me. Like if you can't see what I wrote on the board, or if you can't see the slides very well, I'll fix it if I know there's a problem. Okay. Is this where we should be starting? Somehow I didn't realize that this slide set has the slides for last time and today, so that's useful. And after this lecture, I feel like every lecture after this for the whole rest of the semester is really fun compared to this stuff. So I'm going to let you know out of this section what are the key facts to remember about these different types of metabolism. It's not everything. Definitely not everything. So when we had stopped last time, we had been slogging through some information about these different electron carriers and how they are supposed to be taking electron energy somewhere else in the cell that we consider an electron acceptor. So NADH, FADH2, you could circle those on this slide. They're the most important for this class. Their job is to carry electrons from bonds, from chemical bonds, onto an electron acceptor. They're a transporter for energized electrons. So this group, they transport energized electrons from covalent bonds, like of food, to some type of electron acceptor, like Uber cars, transport people from one place to another. What the electron acceptor is can be in one of two big categories. It's either going to be something from electron transport chain. Sometimes the destination of the electrons is electron transport chain. And if that's where NADH and FADH2 and these others are taking the electrons, we say that that is a kind of respiration. That's cellular respiration process. The other option is if the electron acceptor is something else. Something else that's not part of the electron transport chain. That's what fermentation is. That's what fermentation is in a chemical sense. So this big picture stuff I'm going to leave up here on the board, and as we're talking through what are all these processes that cells use to make ATP, the NADH and the FADH2 are really important to that. The places where they are taking the electrons determines what kind of ATP production is going to happen. Add one more thing onto this. If you're using an electron transport chain, that's going to make lots of ATP. If the electrons are not going to an electron transport chain and they're going somewhere else, and the organism is doing fermentation, that only makes a little bit of ATP. It's not as good energetically. So that's going to become important, and that's something I do want you to know really well out of this section, is these big differences in energy production. Here's your favorite 5 words in the world. Woo! Yay! I think nobody feels that way. I used to, there was a point a few years ago when I realized I have to teach this every semester for like the next 20 years, this topic in a class every semester for the next 20 years. I'm like, that's bad. This is not my favorite. And I think the reason why we don't like to talk about this is because there's so many details and there's so many individual named molecules, and it's just all too much. Cell biology, Jackie, it's all going to be too much. But in here, I want to focus on what does this matter to different kind of microbes. So we're going to assume in this section that every microbe that we're talking about can use glucose in sugar in some way as its food. So glucose is a sugar, glucose is going to be the food source. And there are different ways that different microbes can take glucose and turn it into ATP. So draw an arrow coming out of the word metabolism and make it say here, ATP production. We're going to talk about what are the different ways that a bacterium can take glucose and make ATP out of it, relevant to where are the electrons going. That's going to be the big picture where we're heading with this. Which style do people do? Aerobic. Like we are aerobes. We have to have oxygen to do this well. Microbes can, some of them can do it aerobically, some of them not aerobically. Every organism that is going to burn sugar for fuel is going to start with glycolysis. And I would have you write up at the top of this, turning glucose into ATP always starts here. Turning glucose into ATP always starts with glycolysis. It's the beginning of every type of metabolic pathway that uses that sugar. And then at the bottom, I want us to write what are the big takeaways from what happens in glycolysis. Who's in AMP1? Have you all gotten to this yet? Are you in it like right now? Great. So I know everybody has had some training in this already. So here's the takeaways from what happens in glycolysis. You make 2 pyruvates. These are kind of the outcomes. 2 pyruvates. You make 2 ATPs. whoops, ATP overall during glycolysis. You make 2 NADHs. And this is per glucose. So if you start out with 1 glucose molecule, glycolysis produces in the end 2 pyruvates, 2 ATPs, and 2 NADHs. The ATPs are the best. They are usable for the cell's energy. Okay, so ATPs the cell could go ahead and use. What's going to happen to the pyruvates? What are they for? What does the cell do with that? What is the point of a pyruvate? I can make the dumbest song ever. What is the point of a pyruvate? Say that louder. Yeah, like this still has a lot of bonds. This still has a lot of chemical bonds in it, so it's useful to make more ATP. And one thing that could happen to this is it could go to the Krebs cycle or the TCA cycle. All right, that's in blue. What could happen to what is the point of a pyruvate? That could go into the TCA cycle. That's an option to get more ATP later. What are the NADHs? What should they be doing next? What's their point? Yeah. So an NADH is an uber full of electrons that ought to be taking them to the electron transport chain. So that's going to take electrons to the electron transport chain. And that's takeaways from glycolysis, yeah.
Do you want us to know where it occurs?
Yes, thank you. Yes, where does this occur? Yay, cytoplasm. This is happening in the cytoplasm. It's in the cytoplasm. Glycolysis is happening, floating around in the cytoplasm. That's the kind of stuff I want you to know about glycolysis. It's basically this stuff in the box here. That's for glycolysis. All the different kinds of respiration and fermentation are going to start with glycolysis. That part's always going to be the same to burn glucose. The next thing that normally will happen has to do with those pyruvates eventually are going to make it into what I learned as Krebs cycle. The better name is tricarboxylic acid or citric acid cycle. Those all mean the same thing. And I want you to know kind of the takeaways from citric acid cycle, the same as you know the takeaways from glycolysis. So I'm going to change this box right now, what's in this black box, to make it about citric acid cycle. I'm going to do some erasing. First, let's talk about the location. Where does this happen in people or in the videos? In the mitochondria. Do bacteria have mitochondria? They don't. They don't have organelles. They're prokaryotics. So in bacteria, where does this happen? I don't know. I'm sure this was not in the video. In the bacteria, the only place it can happen is in the cytoplasm because that's what they've got. This is happening in the cytoplasm for bacteria. But in people, it's in mitochondria because we have mitochondria. Bacteria don't have that. The only place they can do this is in the cytoplasm. So that's kind of a big difference between us and bacteria. So at the end of the citric acid cycle, there are some big important products. There is CO2, and I'm writing the numbers that are per glucose, okay? Per glucose, there are going to be 4 carbon dioxides. There are going to be 6 NADH. There are going to be 2 FADH2s. And there are going to be 2 ATPs. Those are the major products. So per glucose. They're also listed over here on this, this thing here. Total yield. And this is like per glucose. 2 ATPs, 6 NADH, 2 FADH2s, and 4 carbon dioxide. ATPs, great, the cell can use those. Cell's going to use those for energy. Where should the NADH Uber and the FADH2 lift take electrons? Where are they going to go? Yeah, they're going to take electrons to the electron transport chain. Where does the CO2 go? No one ever talks about this. How do you know that?
Because we breathe out CO2.
We do breathe out CO2. Like maybe I teach this in my A&P2 class that like when CO2 goes, it just diffuses everywhere. And it eventually gets in the air and it goes everywhere. If you're a little bacterium, E. coli here, and you make CO2, it just diffuses on out of you. You don't even need lungs. It just goes into the air. As fast as you make it, it diffuses out into the air. So we'll just write, it goes into the atmosphere. And sometimes in lab, though, you will see it like make a bubble. If you do the test right, you'll see CO2 coming out in bubbles. Okay, so that's citric acid cycle. And so far, we have all of these NADH and FADH2s hanging around, heading to electron transport chain, which in bacteria looks like this. Now it's their time to shine. I like this so much better than this stuff with the arrows and so on, because at least finally got pictures of proteins that are involved. So this is the electron transport chain in bacteria. If you got slides to write, put up here, this is in bacteria. And it's kind of like our electron transport chain. You've got a bunch of proteins. You've got ATP synthase. The proteins are in a membrane. You've got a proton gradient. That stuff is still the same. But what's different is that this is happening across the cell membrane. This is the cell membrane of a bacterium because it's the only membrane it's got. There's no other choice for where to put this. So it's in the cell membrane, and they're pushing protons outside the cell, technically. So they're doing proton pumping from cytoplasm into the outside of the cell. Is this gram positive or gram negative? How do you know? It's just got this one thick layer of peptidoglycan. So if it was gram negative, this part would still be similar down here. So here's NADH. FADH2, we don't show that, but here's NADH bringing electrons into this system. So if you've got this picture, draw some electrons going this way. Electrons here to here. From NADH into electron transport chain. And then I want you to circle this group of molecules here. Circle all this stuff, if you've got the pictures. Actually, this row of stuff right here. This row of stuff. And call those electron, or call it final electron acceptor. Yeah, the second row. Yeah, not the whole thing. I did say at first the whole thing, but circle this row and call it final electron acceptor. Final electron acceptor. And then circle this row separately, where it says water and these others. And that's the row of waste products right here, waste products. So in electron transport chains, no matter what kind they are, electrons come there, they get dropped off by NADH, and they make their way through the different proteins until they get to the end of this line, and then they're going to get added on to these final electron acceptors. So draw an arrow coming out from here that represents electrons. Here come the electrons. And they are going to go on to one of these molecules that we call final electron acceptors. So electrons leave here, they come out down here. Onto these final electron acceptors. And they join on with these molecules, and then they form a waste product. So as people, we're called aerobic respirers. We have to have oxygen to live because we have to have oxygen to pick up these electrons. That's our only choice. Only oxygen can pick up electrons out the end of our chain, and then we form water as a waste product. That's what aerobic respiration is. So on your overview page right here, if the final electron acceptor is oxygen, so if oxygen is the final electron acceptor, that means it's aerobic respiration. That's what aerobic respiration is. If something else is the final electron acceptor, but there's still an electron transport chain happening, That is called anaerobic respiration. There's two versions of respiration. Both of them use electron transport chain. It just depends, what's at the end of the line here? Is it oxygen or is it something else? If it's something else, that's what anaerobic respiration is. Both of those make lots of ATP. Both of those are a great way to make ATP, except aerobic makes a little bit more ATP than the anaerobic does. I'm going to go back and show something different now. Go back to our friends, the oxygen tubes. You're like, no, tired of the oxygen tubes. It's really important. Let's see. Go find them back here. Here we go. Okay. So think about, this is an anaerobic organism. It has enzymes to deal with oxygen, but it doesn't grow any better where oxygen is than where no oxygen is, because it's not actually using oxygen to make ATP. It's just dealing with oxygen. It grows the same whether there's oxygen or not. A facultative anaerobe is able to use oxygen. It has the ability to use oxygen. It's going to make a little bit more ATP when there's oxygen, and that's why it grows better where oxygen is. It also could use something else to pick up electrons and do anaerobic respiration. That's what it's doing down here. It's doing anaerobic respiration. Up here, it's doing aerobic respiration. It's growing better. The obligate aerobe has to have oxygen, and there's no other option for it. So if it gets too far from oxygen, dead. It has to have oxygen to make any ATP. That's where we'll stop today. Monday, we'll talk about what fermentation is, and then we'll start on the next topic. What is it, viruses? Yay! That's exciting stuff. So fermentation and then viruses on Monday. Don't forget that you have lab questions due Monday as well. Do your lab questions.
I want to get out of intro stuff into like.
Hang on just a second. Let me stop.